Thanks for inviting me here today. As Dr. Willis said, I'm visiting from the Department of Anthropology at the University of San Diego. And I'm going to speak today about some of our earliest human ancestors. And specifically, I'm interested in the point that our hominin ancestors began to eat meat. This is a big change in behavior, and it's correlated with the advent of tool use, and it's also correlated with a significant increase in brain size. I'm an archaeologist, and so I want to talk about some of the archaeological evidence documenting how our ancestors acquired meat and how they competed with other carnivores to get it, and ultimately how this change in diet affected the evolution of human social behavior. So just to give you an outline of what I'll be talking about, I'll start by giving some background about the evolutionary context of meat eating. Meat eating is so important because it's probably what triggered some of the major behavioral changes that took place in our hominin ancestors. Particularly, it's probably responsible for the evolution of cooperative social behavior. And this is a hallmark of modern human behavior. So this is why I'm focusing on meat eating here. I'm going to tell you about some of my own research on the fossil evidence for meat eating in East Africa and how experimental archaeology can be used to interpret this evidence. And I will finish by talking about some of the implications for understanding the evolution of human behavior, and also a bit about some of my ongoing field research. So to begin with, who are our fossil hominin ancestors? We've all probably seen some kind of scenario like this cartoon showing a linear progression from an ape through several phases of development, ending with a modern big-brained technology using human. But this isn't quite how it worked. The pattern human evolution has followed looks something more like this, like a tree, like a branching tree. And the base of the evolutionary tree extends back to earlier than 6 million years ago, when we share our last common ancestor with the apes. And all of the fossil species here collectively referred to as the hominins. These include several different groups. So we have the Australopithecines, Paranthropus, and then our own branch on the tree, the genus Homo. And we're the only living members of the genus Homo. Now, the hominin evolutionary tree is constantly being refined, and pieces are being filled in as we discover new fossils, or we learn more about fossils that we've already found. But one particularly interesting part of the tree is here at about 2.5 million years ago, with the origin of the genus Homo. So the earliest species belonging to the genus Homo was first discovered by Lewis and Mary Leakey right here at Olduvai Gorge in 1960. Olduvai Gorge is in Tanzania. And they named this species Homo habilis, or handyman, because it was thought that this was the first species to make stone tools. So in their 1964 paper announcing the species, they argued that it fit 3 key criteria for belonging to the genus Homo. It had upright posture, which is evidenced by some of these foot fossils that they found, and it had the dexterity to fashion primitive stone tools, and it had a large brain. And paleoanthropologists were reluctant to accept the earliest finds by the Leakeys as a new species because they were pretty fragmentary, but some of the more, later on, some more complete specimens were found at Kubifora, including this skull, KNMER 1813, which is one of the nicest skulls that's been found of Homo habilis. So Homo habilis today is generally accepted to be ancestral to later species in the genus Homo, including Homo erectus. However, there's some variation in the fossils that date to this period, and they may actually represent more than one hominin species. So for this reason, sometimes people prefer to refer to these fossils simply as early Homo rather than Homo habilis. So this is the famous Lucy skeleton, Australopithecus afarensis from Ethiopia. And this is the species that is likely ancestral to the genus Homo. Now, fast forward 1 million years later to Homo erectus, and we find something very different. So this specimen on the side of the screen is the Turkana boy skeleton. This is the most complete specimen that we have of Homo erectus. It's from near Lake Turkana in Kenya. So during the transition from Australopithecus to Homo erectus 1 million years later, there were some important anatomical and behavioral changes that took place. So there was a shift to dedicated bipedalism. So in other words, early members of the genus Homo began walking on 2 feet and only 2 feet. Brain size increased, and also body proportions became more modern. And you can see some of these differences here just by comparing these two skeletons side by side. So here we have a time scale to put this in perspective. So the origin of bipedalism dates back to about 6 million years ago. But it's not until about 2.5 million years ago, with the emergence of the genus Homo, that we see this shift to dedicated bipedalism. Now, although members of the Australopithecines and earlier hominins were bipedal, they retained some more ape-like characteristics, including long arms and more ape-like body proportions, which you can see here comparing this Australopith skeleton to a chimpanzee and a human. And these are adaptations from moving around in the trees. Now, it was early members of the genus Homo that were the first to shift to dedicated bipedalism. So the question then is, why did they start walking strictly on 2 legs? And the reason is thought to be climatic change. So beginning around 5 million years ago, sub-Saharan Africa underwent a cooling and drying phase. And this is what led to the spread of the savanna grasslands that we see in East Africa today. You know, Australopithecines lived in forest habitats, and they relied on woodland plant food. So for them, this meant that their food resources would have become more scarce, and this would have required that they have to range further between food patches every day and expend more energy trying to find food. Now, it turns out that walking bipedally actually uses less energy than walking quadrupedally. So bipedalism probably aided hominins in finding food during this climactic shift, allowing them to more easily move between these food patches that were spread further apart. But bipedalism wasn't the only major transition during this period. Brain size also became larger. So where Australopithecus afarensis had a cranial capacity of 450 cubic centimeters, the brain size of Homo erectus had increased to around 900 cubic centimeters. And for comparison, the modern human average is about 1350. Now, it turns out that brains are actually extremely energetically costly organs to run. They account for between 16 to 20% of our daily energy usage. And if we looked at the amount of energy usage by unit weight, the brain uses roughly 16 times as much energy as muscle tissue does. So it's a very costly tissue for us to maintain. So for humans, this means we use a very large share of our daily energy budget just to feed our brains. For early hominins, an increase in brain size would have required that they adopt a diet with enough calories and enough nutrients in it to meet these new energy demands. So Australopithecines and earlier hominins probably lived in forest environments that may have looked something like this. This is a photo that I took in western Uganda last summer. And there are plenty of plant foods for a primate to eat in an environment like this. But beginning around 5 million years ago, the spreading of the grasslands in East Africa would have limited the amount and the variety of edible plant foods available to hominins. And fossil evidence suggests that different lineages of hominins coped with this environmental change in different ways. So for example, the Paranthropus lineage was a dead-end branch of the human family tree that lived alongside members of the early genus Homo. Paranthropus coped with this change in environment anatomically by evolving specializations, including huge teeth and huge jaws that enabled them to subsist on the more widely available but more difficult to chew foods like seeds or plant roots or tough fibrous plant material. Now, early genus Homo took a different path and began to exploit some of the new resources that became available in these new grassland environments. So as it turns out, the spread of the grasslands also led to an increase in grazing mammals like antelope. And animal tissue is very dense in calories and nutrients. It's far denser than most plant foods in these nutrients. And so hominins who were capable of exploiting these antelope on the African savanna would have had an advantage. Now, evidence suggests this is the strategy followed by early Homo. So Homo habilis, pictured here, had smaller teeth and more delicate jaws that were not adaptive for chewing tough plant material. But one thing that Homo habilis did have was tools. Now, the emergence of the genus Homo is associated with the earliest stone tool industry. It's been called the Oldowan stone tool industry after Olduvai Gorge, where these tools were first discovered. This is one of the earliest Oldowan tools. It dates to 2.6 million years ago, and it comes from a site called Gona in Ethiopia. And also associated with Oldowan tools at several archaeological sites are fossil antelope bones with butchery marks on them. So this indicates that hominins were butchering these animals, and presumably they're butchering them because they're eating them. So these changes in the brain and body size of early Homo compared with their Australopithecine ancestors suggest an increased energy requirement, especially by the time we get to Homo erectus. And increased energy expenditure would have required a higher quality diet, and meat may have been an important component of this. So with early genus Homo, we've got this really significant shift to include including more nutrient-rich foods in the diet, like meat. And these are foods that are being acquired with tools. So this initial increase in brain size, and at the same time, the shift to a higher quality diet would have really set the ball rolling for additional behavioral evolution, with big brains producing more complex foraging behaviors to improve diet, which in turn would have spurred additional brain evolution. So eating a high quality diet is closely linked with the increase in brain size throughout human evolution. Now, we know that early hominins were eating meat because there's archaeological evidence for it at several sites in East Africa where there are dense concentrations of bones and stone tools. These sites indicate that hominins were returning to the same place on the landscape to process carcasses. But just because hominins began including larger amounts of meat in the diet doesn't mean that they turned into carnivores. What we're talking about is a gradual shift from a more ape-like diet, which included more plant foods, to a diet that includes more nutrient-rich foods found in savannas. And these are foods that are harder to acquire. Meat was one of these foods, and meat is the one that leaves the mark in the archaeological record. So bones survive in the archaeological record, and plants usually don't. So it's actually much easier to find direct evidence for meat eating. But plant foods probably were a very important part of the hominin diet, just as they are in modern hunter-gatherer diets. Some of the plant foods that, and other foods that hominins probably ate included wild tubers, like modern hunter-gatherers eat today in Africa. This is a Kung San man in the Kalahari of South Africa harvesting a wild tuber. And tubers are pretty common in grassland environments, and so hominins also probably would have exploited this resource. Also, they may have eaten a number of other plant foods or gathered foods like berries. These are Kongolobe berries that are a staple in the diet of the Hadza hunter-gatherers in Tanzania today. The Hadza also gather honeycomb. Honeycomb is high in calories, and so if you can get at it, it's a great food resource. So So hominins probably were eating a lot of plant foods, but plants don't survive in the archaeological record for us to kind of find a lot of evidence for. One way, though, to get at evidence for plant eating is by looking at use wear on stone tools. So using stone tools for different functions can leave microscopic wear patterns on the cutting edge of the tool. So to try to figure this out, what were hominins doing with these tools? Some of my colleagues and I have made our own stone tools, and we used them to process different kinds of food, and then we compared the wear patterns on our tools to wear patterns on tools from different Oldowan archaeological sites to see if they resemble any of the patterns on our tools. So here are some of my colleagues cutting grass with some stone tools that we've made, and these are some of the Oldowan tools that we made. And when we analyzed the use wear in the archaeological assemblages, what we found was it looks like hominins were using stone tools for a variety of different purposes. So they were cutting wood with these tools, they were cutting grasses and reeds, they were butchering animals or cutting meat, and they were peeling wild tubers. So the use wear that we found on these experimental assemblages looked like what we see in the archaeological assemblages. Why would hominins be cutting wood? Maybe sharpening sticks to help them dig tubers out of the ground. So hominins were eating plant foods as well. We just don't see as much evidence for it in the archaeological record. So this is just to keep a little bit about plant foods in mind, but I'm focused here on meat because this shift to include more meat in the diet probably had a major effect on hominin social behavior. Now, while we do know that hominins were butchering animals to get meat, there are some things that we don't know. And these are things that have important implications for hominin behavior. First of all, we're not sure how these animals were acquired, whether it was through hunting or scavenging. Scavenging is a much less risky activity, and hunting is more dangerous. So if hominins were hunting, this means that they were probably cooperating as a group. We also don't know how much meat they were getting. So if you're getting scraps of meat, that's probably not something that's very socially important because it's hard to share a scrap of meat with somebody else in your group, right? But if they were getting big packets of meat, this would make it a shareable resource. So that would be something that's important to know. We also don't know how they interacted with other carnivores in their environment. How did they compete with carnivores to access meat? So speaking of other carnivores, the African savanna was a much more dangerous place 2 million years ago than it is today. There were more species of carnivores, and hominins hadn't reached their modern body size yet. They were about 3 to 4 feet tall. So the savanna would have been a much more dangerous place for a hominin 2 million years ago. So these are the modern large carnivores of Africa. We've got the big cats, leopards, lions, and cheetahs. In East Africa, there are 2 species of hyenas, spotted hyenas, which are the larger species, and also striped hyenas. We have hunting dogs, which don't have a large body size, but they are somewhat intimidating because they hunt in packs. So these are the modern large carnivores, but in addition to the 3 big cats that we have today in East Africa, during the Pleistocene, there were 3 species of saber-toothed cats that are now extinct. These include Dinofelis, Meganterion, which had the largest canines, and Homotherium, which is the largest bodied saber-toothed cat. Its body size was even larger than a modern male lion. Also, Homotherium, we know from fossil evidence, appears to have specialized on taking juvenile proboscidean prey. So there are these 3 big cats, and also there was a species of giant hyena called Pachycracuta, and this is in blue here, showing the body size of Pachycracuta compared to the size of a modern spotted hyena, which is the largest extant spotted hyena. So these carnivores would have posed a significant threat to hominins. And when hominins began to include meat in their diets, they would have had to evolve strategies to compete with them. And we know that hominins sometimes ended up on the menu themselves because we've found hominin bones with carnivore tooth marks on them. So we know hominins were eating meat starting about 2 million years ago. They were running around bipedally on the savanna using their big brains and their stone tools to get meat. And they were competing with large carnivores over access to meat. And this is where my research kind of comes in. I want to know how exactly they obtained meat and what this may have meant for the evolution of modern human behavior. So back to my research questions. I want to know how hominins, or did hominins have early access to carcasses? And if they did they acquire these carcasses through hunting, or did they gain later access to carcasses through scavenging from other carnivore kills, or maybe it was some combination of these strategies? I want to know how much meat they were getting, and was it a very important part of their diet? And I want to know how hominins competed with carnivores in their environment. So overall, how did they fit into their broader ecology? So to answer these questions, I've examined patterns of damage, so looking at tooth mark damage and cut mark damage on fossil bones from 2 Oldowan sites. And to help me interpret these damage patterns, I've conducted experiments with modern carnivores to model how they actually damage bones. And in this analysis, I looked at the frequency of tooth marks in my models and compared them to the fossil assemblages. And one of the methods that I've applied is to use GIS to visualize patterns of bone damage. So I'll tell you a little bit about that in a moment. So when we're thinking about scavenging, this activity could take a few different forms that are actually behaviorally quite different. The one version of the scavenging hypothesis views hominins as passive scavengers. So they're waiting in the bushes until the lions and the hyenas are completely finished feeding before venturing out to eat whatever scraps are left over. This strategy is safest, but potentially wouldn't yield enough meat to make it a very dependable strategy. So on the other hand, the more aggressive confrontational strategy where hominins may have actively scared off carnivores from their kill right after the kill was made would have potentially yielded larger quantities of meat. And this more active strategy, it's a more dangerous strategy, but potentially would have had a pretty high payoff. So how would hominins have done this? Well, they would have had to throw things and make a lot of noise, something like what's going on in this photo here, or this image. But importantly, single individuals would probably not be doing this. This is the kind of behavior that would have been a cooperative behavior. So my study has focused on the bone surface damage on fossils, because bones can give us all kinds of information about what hominins were doing and what carnivores were doing in the past. So carnivores modify bones in several recognizable ways that give us evidence about what they were doing. They might transport bones away from a kill site. So hyenas are famous for doing this. They might rip a limb off of a carcass and transport it back to their den. The carnivores might differentially consume or destroy different portions of bones. So for example, hyenas also tend to destroy the ends of bones, because the ends is where the grease is, and it's easier to crunch off the end of a bone. But probably the most distinctive signature that carnivores leave on bones are tooth marks like these. These are modern tooth marks, but these types of marks actually survive on fossil bones, and they're visible on fossil as well. So one of the main things that affects scavenging opportunities that might be available to a hominin is the adaptation of the initial carnivore consumer. So hyenas have incredibly strong teeth and jaws that are specialized for bone crunching, and so they're able to more fully consume carcasses. But lions, on the other hand, are flesh specialists, and so they typically don't break bones or consume the marrow. They're really focused on the flesh. And saber-toothed cats were probably very similar in this adaptation, because their large canines would have been at risk of breaking if they really tried to use them for crunching bone. They're actually relatively fragile, these canines. So they're flesh specialists. So carnivores leave marks on bone, and humans also leave distinctive marks on bones when they butcher them. So it's a little bit hard to see because the light is so bright, but there are, this is what cut marks look like. Maybe this one is a little bit easier to see. These are what cut marks look like on bone that's been butchered using stone tools. And these bones here are from an experimental collection of wildebeest bones that were butchered using stone tools. So we find these kinds of cut marks in archaeological assemblages too. Also, when humans process carcasses thoroughly, they'll often use a hammerstone to break open the limb to access the marrow. And this activity leaves a distinctive notch feature along the edge, along the fractured edge of the bone adjacent to where the point of impact was. And then also, there's usually, if you look at the inside of the bone, so this is the same piece, looking at the notch from the outside and looking at it from the inside of the bone cavity, there's usually a flake that's taken off along the inside of the bone cavity. And these are features that are that are good evidence and very distinctive of hammerstone percussion, or really what they're evidence for is evidence of dynamic loading. So you're hitting something in a dynamic way, and the bone is breaking in this pattern versus the static loading that a carnivore jaw would produce, so pushing down in a static loading kind of way. So these are very typical of hammerstone damage to long bones. So I've analyzed fossil assemblages from 2 Oldowan sites in East Africa to look for these types of bone modifications. And these sites date to roughly the same time period that the genus Homo appears in the fossil record. So I've looked at a site called Kangera, which is located on Lake Victoria in western Kenya, and then also a site called FLK Zinj, which is one of the sites at Olduvai Gorge in Tanzania. So FLK Zinj is actually one of the best studied early hominin sites. Its technical name is FLK Level 22 from Bed 1 at Olduvai Gorge. And the site was originally excavated in 1959 by Lewis and Mary Leakey after their discovery of this skull here. And this is the skull that they named Zinjanthropus, and so the site is often called FLK Zinj. This species has now been renamed Paranthropus boisei. FLK, by the way, stands for Frida Leakey Karongo. Frida Leakey was Louis Leakey's first wife, and so he named the gorge, that part of the gorge, after her. Here's Leakey with the original Zinj skull. And this site dates to 1.8 million years old, and it contains thousands of fossil specimens and Oldowan stone artifacts. Many of the bones show evidence of butchery marks. So you can see these butchery marks here on this specimen. Now, several years after the Leakeys discovered the Zinj skull, they found the first specimen of Homo habilis in the same level nearby. It was this jaw, the OH7 jaw, or Olduvai hominin 7. And so it's thought that early Homo and not Paranthropus was the maker of the stone tools and was responsible for butchering the animals at the site. Now, because the collection from Olduvai is so large, and because it's really exceptionally preserved, this collection has been studied a lot, probably more than any other site, for trying to understand evidence of hominin behavior. But actually, there have been some vastly different interpretations of this evidence by different researchers. So the first researcher to do a systematic examination of the carnivore tooth marks on the FLK Zinj fossils was Robert Blumenstein. He was at the time at Rutgers University. And he used modern carnivore feeding experiments to try to interpret the archaeological assemblage. So Blumenstein conducted his experiments mostly with hyenas, and he presented bones with flesh on them to simulate carnivore first access. So that's these carnivore first experiments. And then he also presented hyenas bones that had the flesh removed. These are the carnivore last experiments, and this would simulate hominin primary access and then carnivores scavenging from something that a hominin had already butchered. And so he compared these models, and looking at them, it was clear that in these models, in the carnivore first experiments, more than 75% of the time, there were tooth marks on the limb bone shafts. But when, in the carnivore last experiments, when carnivores were presented with bones that were already defleshed, they only tooth marked the limb bone shafts between 5 and 15% of the time. Now, when Blumenstein compared the FLK Zinj assemblage to these experimental models, he found that 59% of the limb bone shafts were tooth marked. This doesn't really resemble either model, but it's closer to the carnivore early access model. So this finding seemed to indicate that carnivores had relatively early access to these bones. But also, one thing is that there is relatively high, a high frequency of percussion marking on the bones. And this indicates that hominins were involved in breaking open the bones for the marrow. So to explain these different lines of evidence, Blumenstein described a 3-stage model where he proposed a scavenging niche for hominins. So he argued that the high frequency of tooth marks on the limb bones indicated primary access by lions. And this is because lions are flesh specialists. They don't have adaptations for crunching the bone like hyenas do. So if lions had first access, the marrow would have still been intact in the bone for hominins to gain secondary access to. And so then this second stage that he proposed is a secondary access by hominins. So hominins then were able to break open these bones and access the marrow, and this would explain why there's hammerstone damage on these fossils as well. And then he proposed the final stage of scavenging by bone-crunching hyenas to account for the low proportion of limb bone ends in the assemblage. The assemblage mostly has the shaft parts of the bones, but not the ends. So hyenas may have scavenged the ends. So more recently, other researchers have argued against this 3-stage scavenging niche model for 2 main reasons. So a Spanish research group led by Manuel Dominguez Rodrigo has analyzed or reanalyzed the tooth marks at FLK Zinj, and he's claimed that many of the marks that originally Blumenstein assumed to be tooth marks may have been actually produced by fungus growth or some other kind of biochemical process. And Dominguez Rodrigo's group did some experiments where they let fungus grow on bones, and these are some images of that. And they've argued that the tooth pits that fungus, or sorry, the pits that fungus can create on bone, something that looks like this, can actually look like a carnivore tooth mark. So these are actually some photos that I took of the FLK Zinj bones that I do think could be showing the result of some kind of biochemical damage or fungal damage to bone. The tooth mark frequency that Blumenstein originally calculated for FLK Zinj may not actually be as high as he thought because of this fungal damage. Now, a second argument against this 3-stage model is that the original study didn't incorporate any of the cut mark evidence. There's cut marks on the bones as well. And about 15% of the bones at FLK Zinj have cut marks on them. And this is a similar frequency to what's been documented with modern butchery experiments. About 15% of bones show cut marks when humans have first access. Also, of the bones that are cut marked, about half of them are meaty portions that are early in the consumption sequence. So they're portions that humans would go for first because there's a lot of meat on those particular bones. So Dominguez Rodrigo reinterpreted the Zinj site as a place where hominins had early access to carcasses rather than a site where hominins were scavenging from carnivores. Now, one of the goals of my study was to reanalyze the FLK Zinj assemblage using new methods to see if I could help solve this debate. One of the problems with studies trying to interpret hominin meat eating really is that we've been basing all of our interpretations on this single site, on FLK Zinj. And research has really focused on Olduvai because the fossils are so well preserved there. But we really have no way of knowing if this site represents a typical pattern of hominin behavior or how variable hominin behavior might have been until we are able to analyze additional sites. Now, the problem is we just don't have many sites from this time period, but the fossils that we're finding at Canberra are giving us a really great opportunity to compare with Olduvai. Canberra is in many ways similar to Olduvai. The sites are similar in age, and Canberra dates to about 2 million years old. The sites are similar in size, and they have roughly the same number of fossils and stone tools. But there are some interesting differences. So the Zinj assemblage from Olduvai is mostly made up of antelope fossils. Both of them are. But the antelope fossils from FLK Zinj are mostly large size, like wildebeest-size antelope. The antelope fossils from Canberra are mostly small-size antelope, more like the size of a gazelle. And also the environments have been reconstructed as being somewhat different. So FLK Zinj has been reconstructed as a woodland environment, which may have been similar to this modern habitat that I'm showing you. This is Nairobi National Park in Kenya. And the ancient habitat at Canberra has been reconstructed a bit differently. It's been reconstructed as more of an open grassland, similar to this image. This is Ruma National Park in Kenya, and it's actually not far from where Canberra is. So Canberra is located in western Kenya on the Homa Peninsula of Lake Victoria. And you can see in this close-up, here's the peninsula, and here are the exposures at Canberra. This is a Google Earth satellite image, and this is literally how we find fossil exposures, is look at the satellite, and the exposures are light-colored. They're light-colored because they're erosional areas. There's not vegetation growing in those areas. This is Homa Mountain. It's an extinct volcano. So the word Homa actually is the word for fever in the local Luo language that's spoken in that part of western Kenya. And they call it the Fever Peninsula because there are a lot of mosquitoes there. So actually there's a lot of malaria there. And so we never forget to take our anti-malarials when we're in the field. This is what it looks like at Canberra. It's extremely rich archaeologically. There are a lot of fossils eroding out on the surface. And in fact, there are other deposits all around the peninsula that we've identified in surveys, and we're just starting to explore some of those other places, but this is the Canberra area. And this is what our main excavation looks like. We've got 5 excavations going, and this is our largest one, excavation 1. The excavation goes pretty slowly and carefully. The sediment is really hard. It's kind of like digging through cement. Here are some of the artifacts and fossils in the ground. I'll try to point some out. This is a pelvis bone. This is the hip joint of an antelope. Here's a jaw of an antelope, some other antelope teeth, and other pieces of bone. And then we have stone tools that are mixed in along with these fossils. So the fossils that are preserved at Canberra are mostly antelope and zebra. There are also pigs, birds, crocodiles. There's one hippo that has cut marks on it. And the antelope are mostly small antelope, like I said, and of those small antelope, about half of them are juveniles. Here's a sample of what some of the limb bones look like. These are all antelope limb bones. And then there are a variety of different types of damage on the Canberra bones that show evidence of hominin and of carnivore activities. So we've got hammerstone-produced percussion notches like these. These are percussion pits. There are carnivore tooth marks. These are tooth marks. And then this one is a carnivore tooth mark that's overlying a cut mark. So this is a pretty neat fossil, and this indicates, at least in this instance, that hominins had stripped the meat off of the bone prior to carnivore gnawing. Here's some of our finds from Canberra recently, our 2015 field season. This is an antelope scapula, so the shoulder bone, and there are Oldowan flakes lying right next to it. And this was kind of an exciting find. You can't see it in the photo, but there are actually butchery marks on that scapula. So this is literally the remnants of a hominin dinner right here from 2 million years ago. We also found some nice Oldowan tools like this one, and this is an elephant tusk. And so this gives us some idea of the other kinds of animals that hominins were sharing their environments with. This is another one of our recent finds. So in the center of the square right here, this is a cobble that looks like it was used by hominins as an anvil for breaking bones. So we've labeled it all around the anvil, but there are shattered pieces of antelope long bones all around this anvil. So also many of these little fragments have evidence of hammerstone percussion marking on them. So this is definitely a place where hominins were processing bones for marrow. Here's one of these little fragments close up, and if we zoom in, we can see that there's clear evidence of percussion marking on these bones. So these are some of the types of things that we're finding at Canberra. Now, in order to interpret these fossil assemblages, a main part of my research has been to try to build experimental models of the types of bone damage that carnivores create when they gnaw on bones, and the types of damage that humans create when they butcher bones. This is my colleague butchering a goat limb with a stone tool in one of these experiments. Now, since hominins were competing with carnivores in their environment over access to meat, it's really important to also understand the signature that carnivores leave in the archaeological record. So I focused a lot of my research on carnivore feeding experiments, and I've conducted experiments with several different carnivore species. And my aim was first to increase the sample size of the experimental models that were available. So there have been some feeding experiments done, so like the one with hyenas that I mentioned by Blumenschein, but most of these experiments have focused on hyenas, not other carnivore species, and they haven't been very extensive. And then my second aim was to see if I can differentiate patterns of gnawing that different carnivore species produce. So when I'm looking at a bone that's been gnawed, like this one, can I tell if it was a lion or a hyena? So I conducted some of my experiments with wolves at the Wolf Conservation Center in upstate New York. The wolves here are being prepared for eventual release into the wild, and so they're kept under more natural conditions than typical zoo animals. So rather than being fed zoo chow, they're fed a diet of whole carcasses like they would find in the wild. These carcasses are mostly chickens and roadkill deer that are donated from the Department of Transportation in New York State. And it's important that for our research that they're eating whole carcasses, because this means that they encounter fur and tendon and all of the things that come with a whole carcass. And so their gnawing behavior should be similar to what wolves do in the wild. Our sample size from this study was over 1,300 bones. There's a lot of roadkill deer in New York State. And so this actually makes this the largest experimental bone collection that's been published so far. So I've also conducted feeding experiments with big cats, with felids, at the Carolina Tiger Rescue in North Carolina. And I've mostly worked with tigers at the sanctuary, but there are also 3 African lions that were part of the experiments. This is Sebastian the lion. He happily gnawed on bones for me and participated in my experiments. So these cats are fed whole carcasses also for our experiments. And what we would do is we'd put a carcass in the enclosure for about 24 hours, let them gnaw on it or do whatever they're going to do, and then after about 24 hours, we'd send in the keeper, and the keeper would collect whatever was remaining. So to analyze the bone damage in these experimental collections and compare them with the fossil collections, I've applied a relatively new method using GIS. Now, if you are familiar with GIS, you'll know it's a mapping software. But rather than mapping landscapes, what I have essentially done is create small-scale maps of bones. And I plotted tooth marks onto them. So essentially, the bone is my landscape here. So I've used GIS to record the location of different types of bone damage on each element. So looking at tooth pits, tooth scores, cut marks, and percussion marks. So each tooth pit, for example, is plotted onto the template, like I'm showing you here, and then I can create a composite record of the distribution of tooth pits for each bone. So this one that I'm showing you here is a composite plot of all the tooth pits on all of the left femora from my experiments feeding wolves. And then also I can do the same thing for cut marks. So this is the cut mark distribution on an experimentally butchered bone collection. So these are the areas that we tend to find cut marks on the femur. So this is the kind of data that I'm looking for. And then in my analysis, I've used some of the spatial analyst tools in GIS, not just for displaying the location of these bone, the bone damage, but to try to quantify it in a more precise way. And the cluster analysis is a really useful way to try to quantify how modifications are distributed across bones. So what this cluster analysis does is it takes all of the points that I have mapped onto a template, and it measures the distance between each point, and then tries to identify significantly dense clusters of tooth marks. And then on this graphic here, the shaded areas are then showing these high concentrations of tooth marks, and the red areas are the highest. Okay, so that's a bit about the method. Onto my results. These are bones from my experimental studies, and they show the range of damage that carnivores produce on bones. So these are some of the hind limbs in my experiments that were gnawed on by felids, and these are hind limbs gnawed on by wolves. And you can see here that the wolves tend to destroy the ends of the bones more often than the felids do. And particularly, they go for the knee joint. So the bottom of the femur here and the top of the tibia, they tend to chew off. The felids, they leave tooth marks on it, but they don't chew off this area as commonly as the wolves do. So using these experimental bone collections, I've built GIS models that show composite images of where carnivores typically tooth mark bones. So these are some of the different limb bones in my analysis, showing where the wolves tend to gnaw on bones, where they tend to tooth mark bones or chew the bone. So in my study, I've used these models then these models of carnivore damage to try to interpret the fossil bone damage from FLK zinj and Canjera. And here are some examples of, these are fossils from FLK zinj, showing some examples of bone damage. So here's a tooth mark here. Here, this is also a carnivore tooth mark. And then also in the FLK zinj assemblage, there are examples of hominin damage on the fossils. So we've got a really nice flake here from percussion damage. This is a percussion pit. And then also these are butchery marks here. And these are really nicely preserved fossils. These bones are almost 2 million years old, but it looks like these cut marks could have been made yesterday. They're really, they're still sharp inside the mark. So they're really nice fossils. This is the tooth mark distribution on the tibia, so the shin bone of antelope from Canjera, comparing them to zinj, and then comparing with my different experimental models. So this is the carnivore-only experimental model, and this is the experimental model based on Blumenstein's experiments, a hammerstone to carnivore model. So humans first butchered bones and then threw them to the hyenas. That's what this model is. Now at Canjera, the tooth marks on the tibia are randomly distributed. They don't show a clustering pattern, so I'm showing you where the individual marks are here. Also, there aren't very many marks. The tooth marking frequency is low, and this probably indicates that carnivores weren't very active at the site. At FLK zinj, the tooth marks do occur in significantly dense clusters, and these clusters resemble more the hammerstone to carnivore model. So this, and also this pattern is the same for the other bones of the skeleton. So it looks like hominins had first access and carnivores were scavenging here. So looking at the frequency of tooth-marked bones in some of the different experimental assemblages, you can see that The carnivore-only experiments, these are different experiments here, but they all have a pretty high percentage of tooth-marked bones. And then in the green here, these experiments are the hammerstone to carnivore experiments, and they have a significantly lower frequency of tooth-marked bone. And this makes sense because there's not a lot of meat left on these bones to attract carnivores, right? So they're not chewing it. They're not chewing it as much. So when we plot on the tooth-mark frequency from Canberra, it falls within or even slightly below the range of these hominin first experiments. So each dot here represents the frequency calculated by a different researcher. We had 3 researchers do the same analysis, looking at 3 levels from the excavation, so KS 1, 2, and 3. And so there's substantial agreement between different analysts, and also there's a consistency across time in our different levels in the excavation. So these tooth-mark results suggest that hominins were consistently getting access to carcasses before carnivores were. Now we can also look at the distribution of cut marks. This here is the distribution of cut marks and percussion marks on the antelope femora from Canberra. And I'm analyzing the cut marks here using a model called the hot zone approach, which helps to determine if cut marks were made before or after lions had access to them. So it helps to really get at hunting versus scavenging by hominins. So it's been observed in feeding experiments that lions consume carcasses in a patterned sequence. And they don't always consume all of the flesh on bone. So given this, cut marks seen on anatomical areas where flesh usually does not survive lion consumption, this should indicate primary access by hominins. And this has been called the hot zone approach, developed by Dominguez Rodrigo and colleagues. So what I'm showing you here is the surviving flesh on limbs from my own lion feeding experiments. And this shows the pattern that lions typically consume flesh from the upper limb portions, but not the lower limb portions. And you can see here that the upper part, this is the forelimb and this is the hind limb, the upper part where there's a lot of meat, lions consume those portions, but they leave they kind of leave the lower part of both fore and hind limbs alone. So here are Dominguez Rodrigo's cold zones then, mapped over the Canberra data. So the cold zones are opposite the hot zones. These cold zones are areas where flesh remains after felid consumption. And you can see that the cut marks occur in hot zones. And this suggests that hominins had primary access because there would have been no flesh left to remove in these areas if hominins were scavenging from lions. It's a bit about the cut mark evidence. So to summarize the interpretation of Canberra and Zinj based on my analysis, the cut mark evidence suggests that hominins had early access to at least small antelope. There are cut marks in the hot zones. There are low tooth mark frequencies at both sites, and this indicates, and also the random distribution of the tooth marks indicates that carnivore involvement was probably low, and probably carnivores had secondary access. So how were hominins gaining early access to these carcasses with their pretty basic Oldowan tool technology? They may have been hunting the smaller animals. They might have been aggressively scavenging the larger wildebeest-sized animals, so scaring off the carnivores before they were finished. That would allow them to gain early access to carcasses while there was still meat remaining. If hunting for smaller animals is the case, Canberra may be the oldest evidence for hunting in the archaeological record that we have to date. So there are some important implications for understanding the evolution of human behavior here. We know that hominins were foraging in a variety of habitats, from the grasslands at Canberra to the woodlands at FLK Zinj. And they were using stone tools to process both plant foods and animal foods. Now, the use of tools signals a behavioral shift away from a more ape-like subsistence strategy toward using culture to acquire food. Hominins were probably hunting small animals and scavenging the larger ones. Now, hunting is a cooperative behavior, so hominins were almost certainly cooperating in this activity. And because hominins were getting early access to carcasses, they were getting large quantities of meat, and large quantities can be shared. This is a shareable resource. And large-scale food sharing is a behavior that we really only see in humans. So we might be seeing the origin of this behavior with the addition of meat to the diet. Finally, Canberra and Zinj provide evidence that some sites were visited repeatedly over tens to hundreds of years by hominins. And these sites were the focus of butchery activities. Hominins left hundreds of stone tools at these places. So it's likely that these favored places represent something like a home base where hominins would have returned to on a daily basis to share food with extended family members. So some key transitions in human evolution that we're seeing here are using culture to acquire food and cooperation and food sharing. So to end with, I just want to talk a little bit about some of our ongoing research. My work at Canberra was done through the Homa Peninsula Paleo Project. This is a collaborative project that's directed by Tom Plummer from the City University of New York. He was my PhD advisor. And then also a team of researchers from different institutions, including Liverpool John Moores University in the UK and the Smithsonian. We also work closely with the National Museum of Kenya. This is where our expert field excavators are based. So I mentioned that we've started exploring some new sites in the area around Canberra, and last summer we started a new excavation at a site called Nyayanga. It's just a few kilometers away from Canberra. This is what it looks like at Nyayanga. It's Lake Victoria in the background. So the funny thing about the Homa Peninsula and about Canberra is that we've literally found thousands of Oldowan tools and bones that have been butchered by humans, but until last year, we haven't actually found any hominin fossils. And last year, I guess it was a year and a half ago now, while we were surveying at Nyayanga, we were actually thrilled to find a tooth of Paranthropus boisei. So this is the skull that the Leakeys found at Olduvai. It's a species that has these really huge, really easily identifiable molars. You can see this is the tooth that we found, and it definitely looks like a Paranthropus tooth. So we're very excited, and we decided to put in a full excavation this past summer to look for more. This was where we found the tooth. We actually found it under this tree, and this was our lunch spot at Nyayanga. It's the only shade in the area. And someone happened to pick up the tooth while we were eating lunch one day. So we put in, this last summer, we put in a big excavation at Nyayanga. And all of these guys that are helping us out are trained excavators from the National Museum of Kenya. And then some of them are local people that live in the surrounding area. We try to make sure that we have a good relationship with the surrounding community by creating jobs while we're there. And then what we found in the excavation this year was not a Paranthropus, unfortunately, but we did find basically an entire hippo skeleton. And this may look like a pile of hippo bones to you, but it looks like we have the entire skeleton. And here's some close-ups of some of what we found. These are, this is the tooth of the hippo. One of the things that had us really excited about what we found in the excavation this year is that we found some stone tools right in with this hippo skeleton. So we found a hammerstone, and we found 3 flakes. This is one of them, and the flakes are basically embedded right in with the skeleton. So it's just been excavated, so we haven't had a chance to look at the skeleton to see if there are cut marks on it yet. It's in the National Museum of Kenya, and we're going to return over the summer to analyze it further, but I'll be really surprised if we don't find cut marks on it. We have found a lot of bones on the surface of the excavation, so they're not in context, but some of the bones that we found, like this one, do actually have cut marks on them. So geologists are trying to get a good date for the site. It's not been well dated yet, but based on the fauna that we found at the site, it looks like it might be a little bit older than Canberra, so it might be about 2.4 million years old. And if this is the case, this may be some of the oldest clearly cut-marked bone in the archaeological record. So I'm really looking forward to the things that we're potentially going to learn about hominin subsistence and behavior at these sites. So these kinds of projects are never done in isolation. I just want to thank these people and these institutions that I collaborated with and who provided funding for me, and thank you all for coming to listen.